Next is a close-related organism. First, bat. Bat is one of spreader of Nipah virus. The bat spread usually from the fruit bat species. The fruit bat is usually eat fruit, pollen, and nectar. The bat can spread disease in many ways, such as direct contact with the bat, eating food product, and the product are contaminated by the bat. Second, pig. The pig was a second model of Nipah virus that can infect humans. The transmission of Nipah virus can spread to human by human eating the pig. The Nipah virus was recorded spreading at the pig near Ipoh in September 1998. Lastly, human. Humans are the usually victim of Nipah virus. Human can get virus in many ways, such as direct contact with the bat or the pig, which with your body fluid, such as blood, urine, and saliva. Second, consuming food products that are infected to the pig or bats. The food can be contaminated by the body fluid of infected animals, such as palm suck with the infected bat. Third is close contact with the person which is infected with the Nipah virus. The symptoms in humans are mild to severe disease. The symptoms is such as fever, headache, cough, sore throat, difficult to breathing, vomiting, swelling on the brain, and potentially death.
The tools that used to analyze this genome is MEGA-seq. MEGA stands for Molecular Evolutionary Genetic Analysis. It's a free software for conducting statistical analysis of molecular evolution and for constructing phylogenetic trees. MEGA is widely used by biologists in a variety of laboratories to construct species evolutionary histories and infer the extent and nature of the selective forces shaping the evolution of gene and species. MEGA is a comprehensive tool for performing sequence alignment, phylogenetic tree inference, estimating divergence time, estimating rate of molecular evolution, inferring ancestral sequence, and testing evolutionary hypotheses. For methods, laboratory diagnosis of a patient with a clinical history of Nipah virus infection can be made during the acute and covalent phases of the disease by using a combination test of RT-PCR and ELISA. In one step, RT-PCR, reverse transcription or cDNA synthesis, and qPCR are carried out in a single tube by employing sequence-specific primers for the application of the specific target. Genetically modified reverse transcriptase are commonly employed by one-step assay as they tolerate higher temperature necessary for annealing of sequence-specific primers. As one-step assay is preferred when repeated qualification of the same gene is carried out, especially in high throughput and diagnostic application. Another consideration when choosing one-step assay is the quality and scarcity of RNA. ELISA stands for Enzyme Linked Immunosorbent Assay. It's a plate-based assay technique designed for detecting and quantifying soluble substances such as peptide, protein, antibody, and hormone. ELISA performed in 96-well or 384-well polystyrene plates. ELISA used enzyme-labeled horseradish peroxide and alkaline phosphate. In an ELISA, the antigen, which is target macromolecule, is immobilized on a solid surface, which is microplate, and then covered with specific antibody that is linked to a reporter enzyme. The sample that added contained antigen of a particular infection, so we can diagnose that particular disease. The antigen will specifically interact with antibody. After that, we will do washing state for each stage. After that, we have another type of antibody which is specific to the antigen, but this antibody contain enzyme link with it. This enzyme can convert particular substrate to colored product which we can visualize it. Now the antibody along with the enzyme link will attach to the antigen, and then the substrate will give some coloration. Detection is accomplished by measuring the activity of the reporter enzyme via incubation with the appropriate substrate to produce a measurable product. The most crucial element of an ELISA is a highly specific antibody-antigen interaction.